Hello and welcome to the 2016 Croi, which is the Conference on Retroviruses and Opportunistic Infections here in Boston in February 2016. My name is Jeff Taylor, and I'm here today with several people who are working in the cure research arena. Paula Cannon from UCLA,
Nope.
USC, I get it
Thank
wrong
you.
every time.
It's a really bad
University
thing to
of
get
Southern
wrong.
California, I know. Corinne Dubé, who works with the Delaney Cab, and Dr. Warner Green, who is with the Gladstone Institute at the University of California, San Francisco.
Yes.
So welcome all, and thank you for joining us today. And we're going to talk about cure research. So cure research is coming up in the world, and especially at Croi, and we actually have a plenary this year that's going to be moderated by Dr. Cannon. So Paula, you want to tell us some more about what you'll be talking
Sure.
about?
No, I think that's pretty exciting actually, and I was very honestly astonished when I got asked to give a plenary. It's kind of a big deal at Croi, but I'm going to be giving a plenary where I'm going to be overviewing what's happening with, and the contribution that gene therapy is making to cure research. So cure research gets a plenary and gene therapy gets a plenary. Gosh, it's like end of days. So I've been thinking, my plenary is actually tomorrow morning, so I'm sort of a little bit stressed out today, and I've been talking to many people who work in this field about what sort of things should I talk about and what message should I try and get across, because people still say to me, you know, gene therapy for cure, how's that going to work? So there's a lot of non-believers out there, and hopefully I can change a few hearts and minds tomorrow. One of the things I think that's very important for me to convey is that just in the last year or two, there's been a lot of excitement and movement about using gene therapy, not for HIV, but to treat cancers. You know, people are getting increasingly excited about the potential of gene therapy and immunotherapies to cure these, you know, sort of intractable cancers that haven't been treated before. So I think I want to say to people, hey, you know, just as gene therapy is providing some solutions to cancer that people, you know, really hadn't imagined would be possible, you know, have a little bit of faith that maybe we can take some of those same types of strategies and apply them to the problem of, you know, getting, you know, instead of getting a cancer out of the body, the idea would be to get HIV-infected cells out of the body.
Do you want to kind of briefly detail for our audience how that works in a nutshell?
Sure, sure. So there's a couple of different ways you can think about using gene therapy. One way is that gene therapy, so I will say there's not many things that HIV does that I think thank you to the virus, but the fact that it has chosen to infect cells, CD4 T cells, that we can easily take out of the body, that's, I think, an Achilles heel of this virus. So in theory, you can take CD4 T cells out of a person's body, or you can kind of go back up the development pipeline and instead take out some of the bone marrow stem cells out of somebody's body that will give rise to CD4 T cells inside the body. So you can take those cells out of the body, and then a gene therapist can engineer them to give them new properties to basically make them resistant to HIV and then put them back into the body. And so the idea is that you can convert some of a patient's immune system into being resistant to HIV. So that's sort of one general idea. But increasingly, the other idea is that you can also use gene therapy to kind of tweak the immune response so that the immune response that currently fails to clear HIV out of the body, maybe we can do something to those cells so that they can now recognize HIV and help to clear it. So there's these kind of two broad approaches people are taking with gene therapy. And I like to think probably what we're going to end up doing is combining the two together and having this kind of like, you know, full-on attack on HIV using a patient's own cells to basically attack the virus.
So a cocktail for cure, just like we had a cocktail for treatment.
Oh, I think absolutely, yeah. There's enough good ideas out there as well to kind of come up with some combinations.
Well, we're all looking forward to cocktail hour.
It's 8 a.m. It's cocktail hour somewhere.
Exactly. Terrific. Any particular approaches that are going to be discussed or presented at this conference you'd like to talk about?
Right. I don't think there's a lot of gene therapy being reported on. Most of the trials that are ongoing are at that stage where they're still waiting for the 12-month point or longer before they're going to be reporting. There is one poster Wednesday night from Rafiq Sekali's group. He's been analyzing some of the data that's coming through from the clinical trials that have been run by Sangamo Biosciences using the zinc finger nucleases, the genetic scissors that knock out the CCR5 gene as a way to make resistant cells. He's been analyzing some of the patients from that study who seem to be having a good response. Some of the patients, when they've had the treatment and they're taken off antiretroviral therapy, they're actually controlling their virus. They look like elite controllers. And he's been looking at what are the correlates of that. Basically, for the people where that happens, what's different about them. And I think he's kind of, it's pointed to being a feature of what are called the CD8 T cells, so the part of the immune system that can attack and suppress HIV. So I'm looking forward to seeing that poster. That's tomorrow night.
Exciting. And then we've been hearing a lot in the news lately about the CRISPR-Cas9 technology, and not just for HIV, but for cancer and a lot of other, genetic diseases. Is there anything happening in that arena that you can tell us about?
Not that I'm aware of. CRISPR-Cas9 are like another type of genetic scissors. So zinc finger nucleases, these are the ones that are already in the clinical trials because they've been around for a decade. CRISPR-Cas9 is like the new kid on the block. It's kind of easier to make them. In my lab, I have high school students who make CRISPR-Cas9, but so it's been a great democratizing force. Anybody can do gene therapy with CRISPR-Cas9, but of course, to actually develop something and take it all the way to a clinical trial is a lot of work. So I think it's going to be a year or more before we see any of these CRISPR trials coming through the pipeline, but I totally expect there will be.
Yeah, that's exciting. Terrific, thank you. So Dr. Warner, at the opening plenary last night, Kenneth Cole, who is the chair of the AMFAR, the American Foundation for AIDS Research, gave a wonderful plenary talk and talked about how far we've come from the bad old days when Elizabeth Taylor had to harangue Congress just to pay attention to the problem, to now becoming a major force and funder of AIDS research, and you're the beneficiary of one of those grants recently. If you'd like to talk about that and the work you plan to do.
First, I'd like to say I thought Kenneth Cole hit it out of the park last night. It was really an entertaining and moving story. His life story and that of AMFAR's was quite interesting. Yes, we have AMFAR announced early this year a national competition to form the AMFAR Institute for HIV Cure Research. They had no one's quite certain how many universities or consortiums they invited. I know we responded vigorously. A consortium between the Gladstone Institute, the University of California, San Francisco, and the Blood Systems Research Institute. And actually, the process was galvanizing for us. Not only did we come together, but we started working together at a level of effectiveness in the process of creating the grant. And we were lucky enough to emerge as the recipient of the grant. And now we're well engaged in our research. Our studies will focus on toll-like receptors, a shock and kill or a kick and kill type of strategy using toll-like receptors as the kicking agent, and potentially using novel vaccines as a way of eliminating the reactivated cells.
So you have a pharmaceutical partner then as well in that to provide the toll-like receptors? Right.
So yes, we are working closely with IDRI, the Infectious Disease Research Institute in Seattle. They have been pioneers in the use of these toll-like receptor agonists as adjuvants for boosting vaccines. But now we're talking about systemic administration of these toll-like receptor agonists, and there's a safety concern that we have, and we have to go very cautiously because we are triggering the innate immune response, an emergency, a very first-line response in a systemic manner. So our hope is that we can take combinations of toll-like receptor agonists like TLR4 and TLR7 and use reduced doses of the two together and synergize, and so that we can get a beneficial effect. So the idea is to look for a synergistic coupling and then to maybe add to that other components of a cocktail, be they epigenetic modifiers, agents that would increase the transcriptional activity of the LTR, or noise enhancers. So we have many ideas, many experiments to do, and while the grant was a very generous, $20 million grant over 5 years, when you actually divide it up, there's one finds that the money doesn't go very far, particularly when you're doing clinical trials in monkeys and in humans. So, yeah. So we're continuing to look for funds to move this cure initiative forward.
Well, yeah, I think funding is always the big limiting factor in all this research. Certainly Obama a couple years ago announced $100 million towards cure research, and we're finding in the HIV field that that's not new money, that's money that's being redirected towards HIV, and there's a lot of Sophie's choices happening in the research arena as to what gets retained and what gets...
And we are in the midst now of the collaboratory recompetition, so that the collaboratories was created about, it's an idea that emerged about 7 years ago at a workshop in Georgetown. I remember sitting there and listening to Daria Hazuda talk about doing an shRNA screen, and I thought, we're doing the same thing. I said, we've got to start working together. We got to form a collaboratory. And so NIH was at the meeting, and it ultimately led to the idea of forming, and Martin Delaney was at the meeting. And ultimately they were named after him, after his passing. And this is really, NIH is taking the bull by the horns and really pushing the cure agenda through these collaboratories. So we're through the first cycle, and now competition, the programs are being refined. They're being somewhat reduced in size. More mobile teams are being asked for. And so we'll find out in April how the competition has gone.
I think a lot of us have been involved in those applications.
Yeah.
A lot of fingers are crossed. But it'll be interesting to see who gets funded. And, but as you say, there'll be more smaller groups working on more areas. So hopefully it'll yield something exciting. Thank you. And then something kind of near and dear to my heart is the community response to cure research, because at the beginning, when they're proposing trials where people had to undergo, the leukapheresis and all the things you have to do for gene therapy, having to stop their HIV meds altogether to see if these approaches actually worked, is a scary prospect. And the FDA and a lot of the researchers themselves came to the community and said, are people going to sign up for these trials? It's all risk and no benefit. And we have, very easy to take, very easy to tolerate, very effective meds. Why would anybody go off one pill once a day to do these things? But certainly those of us in the community have recognized that there's a huge desire, especially among those of us who've lived for a long time with HIV, to actually see an end to this disease, just as, of course, the researchers do. So we started doing some research, and here with us today is Corinne Dubé, who has helped put together an education initiative to kind of build literacy in the community around HIV called the curriculum. So Corinne, would you like to describe that a little bit?
Thank you, Jeff. Yeah, so the curriculum is an HIV cure research training curriculum. It's meant to make HIV cure science accessible to the community and the HIV research field. It's been a true collaboration between community advocates and scientists. We're basically trying to translate the science of HIV cure research to communities. We're trying to find a balance between portraying accurate science but also making it accessible to people. We just recently received a small grant from the NIH to revamp our curriculum effort. We're going to be focusing on 6 modules, including a basic introduction, stakeholder engagement in HIV cure research, informed consent and ethics, early treatment, and pediatrics. We're focusing on the modules that may be relevant to the road to Durban in IAS. These are the HIV cure research modalities that are more relevant to resource-limited settings. And we have a small funding for that. We're also planning a webinar series in May or June of this year to prepare the road for Durban. We're also advocating for a research literacy zone in Durban.
That'll be at the Global Village,
At
correct?
the Global Village,
in the
yes.
Conference, yeah.
And we're preparing educational materials related to cure research. The goal is to strengthen community participation and community capacity to make decisions about HIV cure research. And we're also promoting the ethical development and implementation of cure research. The concept emerged at Croi 2014 here in Boston, and we were able to launch the curriculum at Croi 2015 in Seattle. So we're really happy with where it's going right now, and we hope that it will continue.
Yeah, well, thank you for all the hard work you put into this, and our community partners, especially at AVAC, Jessica Salzwedel, and a number of us from the Delaney Collaboratories, from the community advisory boards there, have all come together and done this. And of course, the researchers themselves who have very generously donated their time and slides to make this a reality. So, and this can be found on the AVAC website, correct?
Correct. www.avac.org/curriculum. Yes.
Terrific. Yeah, they have a cold cure section on there, so it's a wonderful resource. And the webinars are available as well as the modules online.
Correct.
Yeah. Terrific. And then I mentioned, some of the other community initiatives have been convincing the field that people are willing and able to do this research, and we've wanted to delve into this further to kind of find out what exactly people would be up for, what procedures would they be willing to undergo, what risk would they be able to take. And Corinne, she's a doctoral student, and one of her projects to which she's devoted lots of her own time and resources has been to put together a survey, and we reported some of the results recently. Would you like to talk about that?
Right. So we're trying to understand community perceptions around HIV cure research, perceived risks and perceived benefits. And we're actually finding out that altruism is what promotes participation in cure research, and stigma is what deters participation. So we still have a lot of work to do to reduce stigma around HIV cure, around HIV 30 years into the epidemic.
Yeah, stigma is a huge topic. It was actually the topic of the Delaney lecture yesterday at the opening session for young investigators, and we had a wonderful panel with people from the U.S. and South Africa talking about the huge role stigma has played throughout the course of the epidemic and still does, and being a huge barrier in people accessing and staying on treatment. So it's important now and it will be for the cure as well. And then I think as a follow-up to that, you're planning to do some more detailed research where you delve into kind of the issues that come out and conduct a series of focus groups.
Correct. So we're working with the Martin Delaney CABs in San Francisco, Los Angeles, Seattle, San Diego, Palm Springs, and Chapel Hill to implement focus groups to dig deeper into these factors that would promote participation in cure research. We're also collaborating with the FDA Patient-Focused Drug Development Initiative, and they've provided wonderful feedback. So we hope to have data to share by the IAS meeting in Durban.
Yeah, and as I understand it, I think one of the hopes at the Durban conference is to be able to ask community people attending the Global Village booth to fill out a few short questionnaire about their attitudes as well. I think it'll be really helpful because to date, most of the research has been done here in the U.S. and Europe and Australia, and now we're starting to expand capacity to where it's needed most. So I think it's going to be really exciting. Terrific. Thank you, Corinne.
Thank you, Jeff.
So, Doctors Cannon and Green, do you have anything else you'd like to talk about as we've discussed this that's happening in the field or at the conference?
Well, I just want to follow up on your finding about altruism, because that's so key. I mean, and in the gene therapy field, the clinical trials we do have very small numbers of people, and often it's very tight criteria, and we do struggle to recruit and enroll people. So if you're out there and you're thinking of enrolling in a trial, great. And if you're out there and you've enrolled in a trial, thank you. We recognize it's not easy, and it's potentially it has risk, and just thank you.
I would just make 2 points. One, that despite the incredible efforts of PEPFAR, Global Fund, in terms of addressing the international need for antiretroviral medications, we're only reaching about 14 million. WHO has now said that all 36 million infected individuals should be treated. And just the gap that is created financially between getting from 14 now to 36, it's like the goal lines were moved. It's staggering, and I just don't think that we as a world are going to be able to do that. So in many ways, I view a cure as Africa's best hope, sub-Saharan Africa's best hope to really definitively deal with HIV/AIDS. In that regard, whatever our therapy is, it must be safe, simple, effective, and scalable.
Yeah.
And I think a gene therapy could be that. A single injectable
Down the line, yeah.
could satisfy that scalability requirement.
What about the kill approaches that you're going to be looking at? Do you think those are going to be as scalable? Or?
You know, I think that, yes, I think they could certainly be scalable. I would much prefer a treatment that does not require multiple administrations of therapy, but I think that's almost, with a kick and kill type of strategy, it's almost imperative that we do that. But that's the other side of the coin is that right now, in terms of the number of people cured of HIV in the world, I guess we can claim one.
There's always Timothy Brown.
Right, there's Timothy Brown.
It started the whole thing. Yeah.
So 36 million minus one to go. And it's not easy. This is a very, very tough scientific problem. So I think it's important for people to realize this is going to take time. Don't know how much time. But I do think that mobilizing the best talent, focusing efforts around this activity is going to get us far down the road.
Well, I think one of the points made at the plenary session last night was that you know, AIDS research in general over the last 30 years has yielded a lot of knock-on benefits, that not just for HIV, but for cancer. HCV is a wonderful example of a disease that we can now cure. And it started with the protease inhibitors that we
well, the blueprint for the HCV therapeutics was laid down in all the work with HIV. The difference is HCV doesn't integrate into the genome, and so so cure really does become much more at your fingertips, right. And dramatic results. I'd also say that the great advances in immunotherapy and immune checkpoint inhibitors that's occurring in the cancer field, many of those same agents are agents that we're looking at in the latency field. And so the areas of cancer and latent cure research are actually knocking on each other's doors.
That kind of harkens back to the beginning of the epidemic, actually.
Yeah.
As well. Terrific. Well, thank you all for joining us today and sharing your projects and your expertise and your enthusiasm.
Thank you.
Thank you, Jeff.